No. These four skills are very important in R. If you have learned these four skills, you will be able to perform all type of analysis without any help. So these you must focus on these skills. First is writing. I am telling again, as I know I already have discussed about these skills, but I want to discuss it again and again so that you remember these four things are very important. So writing, reading, script generate and script editing. Now today we again practice on script reading by using a script I have sent in a group about ANOVA. So I copy this complete script from group, and today we will do practice on reading a script, how to read a script and apply this script on another data set. So copy this complete script, and how we convert this script or generate this script for another data set, reading and code generate, or you can say some editing also. Okay, so first our heading is one-way ANOVA with 2K test and bar graph plot. So here we will draw bar graph with 2K test with lettering, as I show you in our previous class. For one-way ANOVA we need a package called stat. So this, for one-way ANOVA we don't need to install any package, but for 2K test also we don't need any package. So these are the basic functions of R in stat package. So we don't need to activate them or library them or install them. We only just need these packages for graphics or our graphs and comparison. We use them for graph and comparison. So we need a package ggplot2, ggtheme, multcompview, dplayer and multcomp. Multcompview and multcomp, these are actually a multiple comparison. Multcomp give us a multiple comparison in the form of a table, while multcomparison view give us a information in the form of a plot. It also can give us information in the form of table as well as in the form of figure. So we first need to install this package. We already have this package ggplot2, so we library it. Now we need to install ggtheme. We don't have ggtheme. First we install this ggtheme. When we run command about ggtheme installation, it show me a red dot. Red dot means our R studio is working. So while it's working, we don't need to run any command, any code, or don't need to change, or we can say we don't need to touch to R. So just wait while it's working. So it show me the final results. Package ggtheme successfully unpacked and MD sums checked. So we need another package called multcomp view. So click on multcomp view and install it. Again it shows a red dot, so we need to wait this. So it installed our package and showing results package multcomp view successfully unpacked. So we need next dplayer, install dplayer. He's showing me you have already installations and you want to restart this before installations. This notification will give us when we have already this package in our R. So I said yes. It will restart and start installation. So you can say it is restarting our R. Okay, now it's restart our R, so we run again dplayer command. Again one or more of the package to be updated or currently loaded. He's saying you have already this package, so we need don't to install this. No. When we say it's no, it start installation. So please you also install this package as well. It's working. No. Another important question is how we know about these packages, or I mean how we know which package is used for, which packages are used for this analysis, or even another analysis which is in your mind, like you have an analysis like principal component analysis in your minds, and you are struggling or trying to do PCA. And the first question in your mind is uh, which type of packages are used here. So how to know about these packages, are these packages in name? Before finding the name of these packages, you will need to first count or known about the step for this analysis. Like for ANOVA, we need to find the step, how many steps are in this ANOVA, and each step you need a different package. So you can get help from ChatGPT. First you find the step and then find packages. So get help from ChatGPT. Type here I want to perform ANOVA with the 2K test please let me know about the packages used for this. You can get a list of packages, and you can also write a sentence. I want to perform a principal component analysis in R. Please tell me the steps, all steps used in for this analysis. He will give you the headings and detail about all steps. This is the only a one-way ANOVA, or in our training sessions different analysis we will perform, but when you are in a real data, or you are performing in your own data set or about your thesis, there are a lot of other analysis which you don't learn in this training. But if you have an idea how to code, read, edit, writing and generate and editing, you will be able to perform those analysis also. So the first step is, if you find a new analysis, first find which packages will be used for this analysis, and find next is find steps for this analysis. So we need two things. These are the very important. One is find packages, which packages are used for this analysis, and second, find steps. Find steps. So these two techniques are very important, or these are two tricks, I can say it as a tricks, how we perform unknown analysis which don't know, or we already have learned about this analysis. So we need to learn two things about this analysis. For example, if you want to perform a PCA, you don't learn before this on R. The first questions should be arise in your mind. One is, which packages are used? No, first question is steps. How many steps are used for PCA, and package, how many packages, or which packages will be used for PCA. So these two questions are very important for analysis which you don't have learned already. If you face a new analysis, you should you must arise two questions. Number one, how many steps are used for PCA or for new analysis, or which packages will be used for PCA or new analysis. So if you find the answer of these questions, you will get the 70% of your analysis, and then you will get help from ChatGPT, and you will get 100% of your script. So we move to next step for our reading. So I library all these packages first. I installed all I have one package left, that is the multcomp. Just click on multcomp, and we installed this. So it is installed. It installed, and now we move to library. So we have all these packages in our RStudio lab. Now we activate all these packages before moving further. So library, 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 again library, and library. So our data set is our packages are activated. All these packages are activated, and some packages are showing warning message or notes, that is deep layer, and the following objects are masked from the package base: intersect, stradiff, stat equal and union. So when we activate dplyr, it will mask or hide the some packages, like as in biological field or biological backgrounds. There are some genes whose suppress or activate, or when activate or dominate, and when they dominate they suppress the they they hide the role of another genes. So dominant genes will appears, and while uh, recessive genes or hide their phenotypes, are not represent or not showing its characters. So similarly, when these are the dominant packages dplyr, when we activate these packages, they will mask or hide the use of other packages, like it's showing these packages will be masked. So when we another package, that is multcomp also is a dominant package and masks some other package, like mass and gassers. So these packages will be masked. So here is a new term. Here is a mean tibble. Tibble is another type of str, but str is a basic function. It also gives us the same results, like you can say first tibble ficus tibble, you can say it gives us the same results, that is showing db1, db2, db3, db4 and chr. db1 mean it is a numbers or continuous variables, while chr mean it is a character. So it will give us a structure of our data set. We can if you here is str is a basic function, so we don't need any package for str. But when we perform tibble, here is the results of strs in similar ways, but here is in the form of columns and here is in the form of rows. str is a basic function of R, while tibble is a for tibbles you need or required a package name dplyr. So tibble is a function of dplyr. So the first step, as we discussed already, for ANOVA we need factors. So change character data to factorial data. So we need to change our data which is in the form of characters, or the data which effect we want to check, we need to convert it into the factors. So convert this data into factor. We here one variable, that is character is species. So we convert species into factor. For factors we use a basic functions, that is factor, and here we learned about how to read a script. Okay, now here is a ficus and dollar sign. Dollar sign mean column variable. It is showing that we are using a file name ficus and its column species. Dollar sign indicate the it is a column or variable name. So here is a less than or minus sign mean equal. So we give a new name to this script. We can manage any name we can take, or we can give any name to this script, but we are here convert a species variable to factor. So as a professional or data analyst, we give you a species name, so that when when we need this variables or scripts, we remember that we save it in the form of ficus column species. So it is very easy to handle. So if you give its name A, B, C, so this is a very confusing and difficult to us to identify which name we have allot this script. So be specific and give it a related name. So here is again factor. It's showing that we want to make a factor of file ficus, and column species. So run the script. He's showing that object I is not found because I select just I, so it run just I, only I. So I need to select all. For all selection, you just click on anywhere in this line and run this script. No, you can again open your file and see, and you can see when we move cursor to near species, and it's showing that it is a character. It is a factor with 3 levels. So after first step is convert dataset into factors. Second step, calculate mean and standard deviations. Mean and standard deviations. So to calculate mean, here we give a new name, mean data, less and minus. It's mean that we are giving this script a new name, that is the mean data, because we are calculating mean. So here we create another file in global environment, and like this its name is ficus. We give new files name mean data of ficus. So grouped by ficus and species. So we mention it that please group our files by species variable. So here is a pipe operator. Pipe operator mean this is the percentage greater than percentage mean pipe operator. Pipe operator is the function of declare, and is a very important functions for combining multiple scripts or multiple codes. It uses the results of one script, one code, and apply on another code like this. When we run the script, here is a complete script or a code. Pipe operator, when we connect through a pipe operator, it will apply these results on next script, on this script. And again we connect this with the pipe operator. It applies about 2 script result on this code or script. So we read this script completely. Mean data mean we are giving it a file new name. You can change here any name or give you any name which you want to do. And less and minus sign mean we want to create an arguments, or we want to give this script a name. Group by, here is an important function used for calculating means. And so group by species. So we command please calculate mean based on only species. Summarize, give us a summary about petal width mean. Here we are calculating only one mean, equal mean petal width. So we also give a new column name in summary, petal width mean equal mean petal width. So it's showing that we want to calculate only just one mean, that is a petal width mean, and other removes, other column will be removed. As in the case of correlation, when we apply -5 or we have given a range, it will just consider only those variables which we have, which we select. So here is a code for mean. Calculate mean, just type mean, and within parenthesis type a variable name or column name on which mean you want to calculate. So next step is standard deviation. Calculate standard deviation. For standard deviation, just standard deviation petal width, similar as like in mean. Here we give a column name mean, and petal width mean. We are going to create a new files. Here when it is created, you can see that here you can see there is a column name or petal width, petal length. We want to create or edit petal width mean. We want to change this column name petal width mean. So we change this after calculating mean. Petal width mean equal mean. Here is a function to calculate the mean. So just click on this and type here column name or variable name whose mean you want to calculate. So next is the standard deviation. Here is a you want to new column, new variable equal by using equal functions. So petal width, and next again pipe operator. Its results will be used by the next script. Range, descending order, petals width, means. So again run this script. Now you get a new files in global environment. Here you can see mean data. Yes, a species variable, we have created a new column, that is petal width mean and standard deviation. So we have calculated mean and standard deviation. Now we apply ANOVA on this data set. Next step is ANOVA. So here is two questions. The one is how many steps are used for PCA and which packages will be used. So here you can see we are performing step by step. We perform first step, that is factor to character, second step mean standard deviation, third step calculate ANOVA. So for ANOVA we use the function AOV. It is a basic function of R. We don't need any package for this. So we want to calculate ANOVA. Petal width, and here is a tilde sign. It is a very important sign. It's showing the effect. It's mean we want to calculate effect of species. Petal width, effect of species. We want to check the effect of species on petal width. This line show that we want to check the effect of species on petal width. Here is our data is equal to ficus. So run this script. You can say another file is created in global environment. Now we have three files in global environment. When you open ANOVA, there is a raw data. This data is not according to our need, so we needed summary, not this complete raw data. Run this next script, that is summary of ANOVA, and you will get summary for this. No, you can see in console we get a summary of our ANOVA results, and it show that it is a here is a p-value. It show that 3 stars. It means our hypothesis is significantly correct, and it show the significant effects of species on variable. So species significantly affect the petal width. When you change species, petal width also change. Now we have calculated ANOVA. Our first task is complete. Now we want to apply 2K test. Remember that 2K test or any post-hack test will be applied on ANOVA result, not mean data, not replicated data. It will be applied after calculating a ANOVA. So if we want to perform a 2K test, you first need the results of ANOVA, and then you want to apply 2K test, because 2K test will be applied on the ANOVA results. Now you can see in here is also 2K HSD. Here is the basic function of R 2K HSD. Here is a basic function of R, and just type ANOVA. Now we want to apply on this file, not ficus file, not mean data. We are going to apply 2K test on ANOVA file. So type here file name, that is ANOVA. Run this, and after run this you can run 2K to get summary of 2K test. Here is important. ANOVA just tell us there is a significant difference or there is a significant effect, but ANOVA don't tell us how many effect, how much effect. Post-hack test tell us how much effect or how much difference. So you can see in the results, in species 1 and species 2 there is a difference 1.08, 1 and 3 there is a 1.78, and 2 and 3 there is a 0.78 difference. So maximum difference was in 1 and 3. Now we compute pairwise comparison using general linear hypothesis test. We have performed two steps, that is one ANOVA and second is Tukey test. Similarly, like in our biological lab, for example, when we are going to perform a PCR or we want to apply a primer, we need a different step. First step, extraction of sample, then extraction of DNA, and then next step is preparing of DNA solution with other chemicals, and then next step is performing PCR, and then next is gel electrophoresis, and after then lab analysis we move to our statistic analysis. So there are different steps for each experiments in lab. Similarly in R there are different steps, different methods to perform an analysis. So we have performed two steps, that is we have calculated ANOVA and we have calculated Tukey test. Now we apply to GLHT. GLHT means general linear hypothesis test. We want to test our hypothesis, either there is a significant difference between them or not. So we used the functions Tukey GLHT, general linear hypothesis test. This function will use a linear hypothesis test to give us its result. So we name this script. Now we apply this on Tukey test, not in ANOVA file. We have created another file named Tukey. No, we similarly as in our biological analysis, biological experiments, we create a next sample, and our next analysis will be next new samples. And similarly as again I will take example of a PCR. First we have a seed or leaf, and then we extract our DNA. So we move to next step, DNA to next step, and next step to another step to gel electrophoresis. So we ignore previous one. So when we have in this our file, first we have a ficus. From ficus we have created mean data. From mean data we have created ANOVA. From ANOVA we have created a Tukey. From Tukey we will create GLHT, general linear hypothesis test. So we apply ANOVA. Here is our data is we apply this is on ANOVA and Tukey test, species column Tukey. So we used combine. Here is ANOVA and Tukey files. So here is a function linfict, that is MCP, multiple comparison. Here is a MCP mean multiple comparison. So run the script. And another you can see another sample is created, or another file is created in global environment. So extract group letter using CLD, compact letter display. Here is another function CLD, that is the basic function of R. Apply this CLD to extract group letters. So run this script. So group letters are extracted. Here you can see, if you click on, you have see raw data, but we don't need this raw data, we needed summary. Now adding group letters to the mean data. We edit these group letter with mean data. We want to combine. Now you have these files, just only pattern width and standard deviation. We want to create another columns, name, group letter, like A, B, C, and so on, based on their variations. So adding group letters to the mean data. Click on No, we have added group letter within main data. Now you can see group letters are added A, B and C on main data. Now if you want to check table and otherwise ignore it. No, we have all analysis. We just need to draw its plot. I hope you will be able to read all this information given in this ggplot too. So these are not difficult for reading, as you already have done practice lot of time on ggplot. So I run the script. Now you can see plots have been created with facet wrap. I deleted here facet wrap. Now you can see you have created a plots with the ANOVA results and ANOVA and 2K test after post-hack test, 2K test and comparison of these 4 species with group lettering. Hope today class will be interesting and you will be understand this. We have just learned just reading and running the script. We read the script and run the script. Now your today task is completely copy the script, Control A and copy and paste it in R. And ask him for explanation. Paste here. Paste it here. And ask to ChatGPT, please explain each and every word of this script, and just read these instructions. What are it's give explanation or explain about this package. So here is a explanation. ggplot2 for creating the complex plot, and he tell you each and everything about in this script. So today your task is just reading this script and understanding this script completely, and you need to learn everything. When you find a new function that is a pipe operator, search it in a pipe operator. It is also give us here about, if not give it's about pipe operator, search here in about pipe operator. So today you will find, or you will need to perform your task to read this script completely, and even each and every word you learn and you will be need to learn. I hope today class you have got it all, and this which I want to teach. I just want to teach how we read script. So reading is also very important. If you have a reading skill, then you will be able to edit any script. If you will be able to read the script, you will be able easily modify the script. You will be able to edit the script, or you will be able to regenerate the script according to for any other data set. Okay, thanks. If you have any question, you can please ask in group or UK. Okay. Have any question? Okay, thanks. Today again I am telling you, today is our topic is just reading, not focus on a complicated script, how I write this script. You just read the script and learn each and everything in this script. Okay, thanks.